The creation of a life form by a similar life is called reproduction. Reproduction is the extension of life of a species at a given time. It is a means of perpetuation of the species and also multiplies their number. By this process, the individuals transmit life to the next generation and thereby ensure the continued existence of living organisms on Earth. If there was no reproduction, life on this earth would sooner or later come to an end. Plants reproduce by several methods. The plants that sexually reproduce have the reproductive structures called the flowers. After completing this digital learning module, learners will be able to explain the pre-fertilization structure and events in a plant, understand the process of double fertilization in plants, express the post-fertilization structure and events in plants, understand the Apomixis and polyembryony. Flowers hold sex organs in a flowering plant. The ovules that develop into seeds are enclosed within an ovary of the flower. The flower is a reduced shoot with the nodes present very close to each other. The different parts of the plant attached to one node are collectively called the whorl. The outermost two whorls are called the calyx and corolla. They are non-reproductive whorls. The inner two whorls are the reproductive whorls and are called anoecium and gynoecium. Calyx. Calyx is the outermost part and most often green in color. The individual units of calyx are called the sepals. It protects the inner whorls at bud stage. Corolla. The next inner brightly colored whorl is called corolla. The individual units of corolla are called petals, which serve to attract bees, birds, etc. that are the agents of pollination. Stamen. Anoecium is the male reproductive part of the flower and stamen is the single unit of it. Each stamen has a thread-like filament at the free end of which there is a four-lobed anther. Carpel. Gynoecium is the female reproductive part of the flower and carpel or pistil is the individual unit. Stamen, Microsporangium and pollen grain. Stamen consists of two parts, long and slender stalk called filament and terminal bilobed structure called anther. A typical angiosperm anther is bilobed. Each lobe has two theca, so they are called dehisces. There are 4 microsporangia in anther, located 2 in each lobe. Structure of microsporangium. Each microsporangium is surrounded by 4 wall layers: epidermis, endothecium, middle layer, tapetum. Tapetum is the innermost layer which has multinucleated cells with dense cytoplasm. It nourishes the developing pollen grain. In a young anther, the center of each microsporangium contains compactly arranged homogeneous cells called sporogenous tissues. Megasporogenesis. The process of formation of microspores from pollen mother cell through meiosis is called Megasporogenesis. The sporogenous tissue of microsporangium is differentiated into microspore mother cell or pollen mother cell. Each microspore mother cell undergoes meiosis and gives rise to haploid microspore tetrad, which dissociates to form 4 microspores on dehydration. Each microspore develops into a pollen grain. Pollen grain. Pollen grain is spherical, shaped, covered by 2 layers and measuring about 25 to 50 micrometer in diameter. Pollen grain represents the male gametophyte. Outer layer is exine that is made up of sporopollenin. It can endure high temperature, strong acids, and alkali and enzyme activity. The exine has prominent apertures called germ pores where sporopollenin is absent. Inner wall of pollen grain is called intine. It is thin and continuous layer made of cellulose and pectin. On maturity, the pollen grain contains two cells, the vegetative cell and the generative cell. The vegetative cell has food reserve and a large irregularly shaped nucleus. The generative cell is small and floats in the cytoplasm of vegetative cell. In 60% of angiosperms, pollen grains are shed at this two-celled stage. In others, the generative cell divides mitotically to form two male gametes before pollen grains are shed, three-cell stage. After shedding, the viability of pollen grains depends on temperature and humidity. In wheat and rice, they lose viability within 30 minutes of their release. In Rosacea, Leguminosea and Solanaceae, they remain viable for months. Pollen grains of a lot of species can be preserved for years in liquid nitrogen, -196 degrees Celsius. Impact of pollen grains on human health. Pollen grains may cause severe allergies and bronchial afflictions. It may cause chronic respiratory disorders, asthma, bronchitis, etc. Pollen grain of Parthenium or carrot grass causes pollen allergy. Pollen grains are rich in nutrients, hence used as pollen tablets for food supplements. Pollen consumption increases the performance of athletes and race horses. The pistil, Megasporangium, ovule and embryo sac. The gynoecium is the female reproductive part of the flower. It may contain single pistil, monocarpillary, or may have more than one pistil, multicarpillary. Fused pistils are called syncarpus and free pistils are called apocarpus. Each pistil has three parts, the stigma, style and ovary. Inside the ovary is the ovarian cavity, locule. The placenta is located inside the ovarian cavity. Megasporangia or ovules arise from the placenta. The number of ovules inside the ovary may be single or many. The Megasporangium ovule. Ovule is a small structure attached to the placenta of locule with a stalk called funicle. The body of the ovule is fused with the funicle in the region called hilum. Hilum is the junction between the funicle and ovule. Each ovule has one or two protective envelopes called integuments. Integuments cover the ovule except at an opening at the top called micropyle. Opposite to the micropylar end is the chalaza, representing the basal part of the ovule. Megasporogenesis. The process of formation of megaspores from the megaspore mother cell is called megasporogenesis. There is a mass of tissue called nucellus present in the center of the ovule. Nucellus cells have abundant reserve food materials. One of the nucleus towards micropylar end differentiates into megaspore mother cell MMC. It is a large diploid cell with dense cytoplasm and prominent nucleus. The MMC undergoes meiotic division resulting in 4 haploid megaspores. Female gametophyte. There are 4 megaspores, out of which only one is functional and the other 3 degenerate. Functional megaspore develops into the female gametophyte or embryo sac. Formation of embryo sac from a single megaspore is called as monosporic development. The nucleus of the functional megaspore divides by mitotic division to form 2 nuclei, which move to the opposite pole, forming 2 nucleated embryo sac. 2 successive mitotic divisions lead to the formation of 4 nucleate and later 8 nucleate stages of the embryo sac. All mitotic divisions are free nuclear type. That is, karyokinesis is not followed by cytokinesis. 6 of the 8 nuclei are surrounded by cell walls and organized into cells. 3 cells are grouped together at the micropylar end, constitute the egg apparatus that in turn consists of 2 synergids and 1 egg cell. Synergids have special filiform apparatus which play an important role in guiding the entry of pollen tube into the synergids. Three cells arranged towards chelazal end are called antipodal cells. The large central cell has two polar nuclei. A typical angiosperm embryo sac at maturity is 8 nucleated and 7-celled. Transfer of pollen grains from the anther to the stigma of a pistil is termed as pollination. Both male and female gametes are non-motile. Kinds of pollination. Autogamy. Pollination within same flower is called autogamy. In open and exposed anthers and stigma, autogamy is rare. In some flowers like viola, oxalis and camelina produce two types of flowers. Chasmogamous, exposed anther and stigma. Clistogamous, closed anther and stigma. Clistogamous flower is invariably autogamous and produces a short seed set even in the absence of a pollinator. Geitonogamy. Pollination between two flowers of the same plant is geitonogamy. There is involvement of pollinating agent. Xenogamy. Transfer of pollen grains from the anther to the stigma of different plant is xenogamy or cross-pollination. Agents of pollination. Plants use two abiotic agents, that is wind and water, for pollination. One biotic agent for pollination is animal. Majority of plants use biotic agents for pollination. Anemophily. In anemophily, the pollinating agent is wind. Plant produces enormous amount of pollen available for pollination to compensate the uncertainties of pollination. Flowers have well-exposed stamens, large feathery stigma to trap airborne pollen grains. Most wind-pollinated flowers contain single ovule in one ovary and numerous flowers packed into an inflorescence, example corn cob. Wind pollination also requires pollen grains to be light and non-sticky. Hydrophily. Pollination occurs by abiotic agent water. Vallisneria, hydrilla and zostera are the common examples for hydrophily. Pollen grains are released into the surface of water and carried to the stigma by air current in Vallisneria. In seagrass, the flowers remain submerged. Pollen grains are long ribbon-like and carried passively inside the water. Pollen grains are protected from wetting by mucilaginous covering. Pollination by biotic agent. A wide number of flowering plants use a range of animals as pollinating agents like bees, butterflies, etc. Insect pollinating flowers are very large, colorful, fragrant and rich in nectar. When the flowers are small, they are present in clusters to make them conspicuous. Flowers pollinated by flies and beetles secrete foul odors. Nectar and pollen grains are the usual floral rewards for insects. In some species, floral rewards are in providing safe places to lay eggs, example, Amorphophallus. A species of moth and yucca plant cannot complete their life cycle without each other. The moth deposits its eggs in the locule of the ovary and the flower in turn gets pollinated by the moth. Many insects may consume pollen or nectar without bringing about pollination. Such floral visitors are referred as pollen or nectar robbers. Outbreeding devices. Majority of the flowering plants produce hermaphrodite flower and undergo autogamy. Continuous autogamy or self-pollination results in inbreeding depression. Flowering plants have developed many devices to avoid self-pollination and to encourage cross-pollination. Such devices are called outbreeding devices. Pollen release and stigma receptivity is not synchronized. Spatial separation of anthers and stigma. Anther and stigma are placed at different positions. Self-incompatibility. Production of unisexual flowers. Pollen-pistil interaction. All the events from pollen deposition on the stigma until pollen tubes enter the ovule are together referred as pollen-pistil interaction. Pollination does not guarantee the transfer of the right type of pollen grain to the right type of stigma. The pistil has the ability to recognize the pollen, whether it is compatible or incompatible. If the pollen is of right type, the stigma allows the pollen to germinate. If it is of wrong type, the stigma rejects the pollen, preventing germination. The ability of the pistil to recognize the pollen by continuous dialogue is mediated by chemical like boron, inositol and sucrose level. Following compatible pollination, the pollen grain produces pollen tube through one of the germ pores. Content of the pollen grain move into the pollen tube. Pollen tube grows through the tissues of the stigma and style and reaches the ovary. If the pollen grain is in two-celled stage, the generative cell divides and forms two male gametes inside the pollen tube. If the pollen grain is in three-cell stage, the pollen tube carry two male gametes from the beginning. Pollen tube enters into the ovule through micropyle and then into the embryo sac through synergids guided by filiform apparatus. Artificial hybridization. This is a major approach of crop improvement program. In this type of hybridization, only desired pollen grain is used for pollination. Stigma is protected from contamination from unwanted pollen grain. Removal of anthers from the flower bud before the anther, dehisces in a bisexual flower, is called emasculation. Emasculated flowers are covered by bags generally made up of butter paper to prevent contamination of its stigma with unwanted pollen. This step is called bagging. If the female flower is unisexual, there is no need of emasculation. On reaching the stigma, the pollen grains put out a tube. This is called germination of the pollen grain. The tip of the tube contains the male nuclei. The tube grows and enters the ovule where it bursts at the tip, releasing the male gamete. One of the male gametes fuses with the egg, the female gamete. The fusion of the male gamete with the female gamete is called syngamy. This results in the formation of zygote, that is diploid. The zygote develops into the embryo. The other male gamete fuses with the polar nuclei. This results in the formation of a triploid nucleus called the endosperm nucleus. Since the process of fertilization involves two fusions, it is called double fertilization. Endosperm. Endosperm develops before the embryo development. Primary endosperm cell divides repeatedly to form a triploid endosperm tissue. Cells of this tissue are filled with reserve food material and are used for the nutrition of the developing embryo. Primary endosperm nucleus PEN undergoes successive nuclear division to give rise to free nuclei. This is called free nuclear endosperm. Subsequently, cell wall formation takes place and endosperm becomes cellular. If we see an example, the coconut water is free nuclear endosperm and the white kernel is the cellular endosperm. Endosperm may be consumed completely during embryo developed, or it may be consumed during germination of seed. Embryo. Zygote is formed and placed at the micropylar end of the embryo sac. It develops only after some amount of endosperm is formed. Embryo development takes place in the following stages: proembryo, globular stage, heart-shaped, matured embryo. Dicot embryo. A typical dicotyledonous embryo consists of an embryonal axis and two cotyledons. Embryonal axis above the cotyledon is the epicotyle that terminates into the plumule or stem tip. Embryonal axis below the cotyledon is the hypocotyle that terminates into the radicle or root tip. The root tip is covered by a root cap. Monocot embryo. Monocot embryo possesses only one cotyledon. In grass family, the cotyledon is called scutellum. It is situated towards one side of the embryonal axis. Radicle and the root cap are enclosed by a sheath called coleorhiza. The embryonal axis above level of attachment of scutellum is called epicotyls. It has the shoot apex or plumule enclosed by hollow foliar structure called coleoptile. Seed. Seed comes from the sexual reproduction. It consists of seed coat, cotyledon and an embryo axis. Cotyledon stores the reserve food material for development and germination. Mature seed without endosperm is called non-albuminous, groundnut. A part of the endosperm retained in mature seed is albuminous. Remnant of the nucellus in the matured seed is called perisperm. Example: black pepper, beet. Ovary. The wall of the ovary develops into the wall of fruit called pericarp. Fruit developed from the ovary is called true fruit. In apple, strawberry, cashew, the thalamus contributes in the fruit formation, which is called the false fruit. Fruit developed without fertilization is called parthenocarpic fruit. Apomixis and polyembryony. Apomixis is very common in Asteraceae and grasses. In this, seeds are produced without fertilization. It is a type of asexual reproduction which mimics the sexual reproduction. Diploid egg cell is formed without meiosis and develops into seed without fertilization. For example, in citrus and mango, the nuclear cells start dividing, protrude into the embryo sac and develop into embryo. In such species, each ovule contains many embryos. Ovule having more than one embryo is termed as polyembryony. Hybrid plants are developed by apomixis to maintain the genetic identity. Let us summarize what we have learned. Typical stamen consists of two parts, long and slender stalk called filament and terminal bilobed structure called anther. The process of formation of microspores from pollen mother cell through meiosis is called Megasporogenesis. Pollen grain represents the male gametophytes. The gynoecium may contain single pistil, monocarpillary, or may have more than one pistil, multicarpillary. The process of formation of megaspores from the megaspore mother cell is called Megasporogenesis. Transfer of pollen grains from the anther to the stigma of a pistil is termed as pollination. Many insects may consume pollen or nectar without bringing about pollination. Such floral visitors are referred as pollen or nectar robbers. All the events from pollen deposition on the stigma until pollen tubes enter the ovule are together referred as pollen-pistil interaction. Triple fusion. The second male gamete is fused with the secondary nucleus to form a triploid primary endosperm nucleus. Since two types of fusion, syngamy and triple fusion, take place in the embryo sac, the phenomenon is termed as double fertilization. A typical dicotyledonous embryo consists of an embryonal axis and two cotyledons. Apomixis is a type of asexual reproduction which mimics the sexual reproduction. It involves the formation of seeds without fertilization. Some angiosperms produce more than one embryo in their seed. This phenomenon is called polyembryony.